Hello everyone and welcome to this Khan Academy tutorial on A-level biology. It's been some time since I last posted a video, but anyways, in this video we'll be looking at the final part of the nervous system and we'll be covering reflexes, synapses and summation. So let's get started. So here are the aims of this tutorial. We'll explain the importance of a reflex and how it occurs, understand what is a synapse and how it functions to transmit impulses, and then understand what are neurotransmitters and describe the types, and finally look at summation and also describe the types. So let's start by defining a reflex. Did you blink? So this blinking is an example of a reflex, and reflexes are described as rapid, autonomic, involuntary responses to stimuli. The aim of the reflex is to protect your body from danger, because imagine if you were watching this video without blinking. Your eyes would dry and that would cause damage. But don't try this, by the way. Because we don't think about reflexes, they aren't coordinated by the conscious parts of the brain, such as the cerebrum. In a reflex action, essentially three neurons control it: sensory, relay, often found in the spinal cord, and the motor neuron. Let's have a look at the common reflex arc that's often tested in exams. So thermoreceptors detect the heat, and the sensory neuron here would transmit the action potential to the relay neuron found in the spinal cord, which provides a response to the muscle via the motor neuron, and the muscle will contract and cause flexion of the hand, which will mean that you move the finger away from the heat source. So now let's have a look at synapses, the good old synapse from GCSE. Synapse is a gap between two neurons, or a neuron and a muscle, also called a neuromuscular junction. We'll learn about neuromuscular junctions later. Neurotransmitters are released in synapse to transmit action potentials, and the neurotransmitters can be excitatory or inhibitory. Excitatory neurotransmitters decrease the threshold potential of the postsynaptic neuron so that action potentials are easier to generate. A common example, which you do not need to know for A-level, is glutamate. Inhibitory neurotransmitters, meanwhile, increase the threshold potential of postsynaptic neuron, so it's harder to generate an action potential. A common example, again, which you do not need to remember, is GABA. So this is an example of a synapse, which you should be very familiar at A-level, and we'll look at this in greater detail on the next slide. So firstly, neurons are named by the neurotransmitters they release into the synapse. So for example, a GABAergic neuron releases GABA into the synapse. A cholinergic neuron releases acetylcholine into the synapse. And this is the neuron you have to know and how this functions. Let's have a look at how cholinergic neurons transmit the action potential to the postsynaptic neuron. So firstly, an action potential arrives at the presynaptic neuron, and this stimulates voltage-gated calcium ion channels to open. And what happens after is that calcium ions enter, so this is called the influx of calcium ions, into the presynaptic neuron. And when they enter, they cause the fusion of acetylcholine containing vesicles into the presynaptic membrane. And when they fuse, the acetylcholine gets released and it binds to the acetylcholine receptor on the postsynaptic membrane. So like I said, acetylcholine diffuses across the synapse and binds to the acetylcholine receptor on the postsynaptic membrane. And this opens sodium ion channels, which is dependent on acetylcholine binding to the receptor. So when the sodium ion channels open, there's an influx of sodium ions into the postsynaptic neuron, which causes depolarization. And if threshold is reached, an action potential is generated. So what happens to the acetylcholine that remains in the synaptic cleft? Well, it gets broken down by the enzyme acetylcholinesterase. And it gets broken down to acetate and choline, and this is reuptaken by active transport. So this process requires energy. And if you remember from the respiration topic, you can see how the mitochondria is also involved in the synthesis of acetylcholine. Acetyl-CoA, which is a respiratory substrate, can bind to choline to form acetylcholine, and again the cycle goes back again. So now let's have a look at summation. Summation, as the word suggests, is the addition of action potentials at the synapse. There are two types of summations. One is spatial, which adds up potentials from different areas around the postsynaptic neuron, and temporal summation, where action potentials are added depending on the frequency. And the benefits of summation is that it allows modulation and differentiation of action potential. So this will allow neurons to determine whether the stimulus is significant or not. So let's have a closer look at spatial summation. So in spatial summation, two or more presynaptic neurons synapse with the postsynaptic neuron. And as you can see in the diagram, there are 4 to 5 inputs. And because neurons can either release excitatory or inhibitory neurotransmitters, the level and the combination of the neurotransmitters will determine if an action potential is generated in the postsynaptic neuron. So this is an example shown in the diagram where 5 inputs together summate and cause an action potential to be generated in the postsynaptic neuron. Now let's look at temporal summation, which is the final part of this video. So temporal summation, like I said before, depends on the frequency of action potential reaching the postsynaptic neuron. The higher the frequency, the more neurotransmitters released by the presynaptic neuron into the synaptic cleft over here. So if there's a lower frequency, the amount of neurotransmitters released may not be enough to generate an action potential in the postsynaptic neuron. Remember, in the previous video I mentioned how the frequency of action potentials determines the intensity of the stimulus. So this is one way of how it determines it. So if there's a higher frequency, the postsynaptic neuron knows that the stimulus is of high intensity. So in temporal summation, the higher the frequency, the quicker the action potentials add up, and this will be enough to generate an action potential in the postsynaptic neuron. So like I said, this will mean that the action potential is more likely to occur. So here's a recap of the aims that we went through in this tutorial. As usual, use them as questions to consolidate your understanding. Thank you for watching, and I will see you in the next video.